Hello everyone, I hope you all are doing well. So, welcome to the Drishti IAS discussion of Mains 2022 questions. Myself will take science and technology, and in this context, I am going to cover three important science and technology questions asked in GS3 paper. I hope those who have appeared in the exam, they have really done well and they were able to write answers. And those students who will be trying for the exam next year, so this particular session is in one way giving you an outline and the way to approach such questions in the examination. Now you know the dynamics of the exam. Every year in the prelims exam and the main exam, the nature of science and technology questions are changing. Same, if you look at last year question paper, questions were not as predicted, right? Questions are linked with current affairs, but the questions are asked in such a way that a candidate need to know all dimensions of the question. So like last year, the Nobel Prize in blue LED light was asked. Then the question was in terms of achievement and development of biotechnology part of it. So those were the questions part of it. Now, if you look at this year question 2022, UPSC again asked questions from current affairs like JWST, James Webb Space Telescope, which was in the news. Very recently it was in the news, and the first image captured by the JWST was in the newspaper. UPSC asked the question on that. But again, it is asking about the telescope, the features, what goal, benefits. So that is being asked. One more question has been asked from health and aspect of this, and in continuation of what UPSC has been asking in the prelims exam, that is based on the vaccine development. That is a question from biotechnology. So vaccine development and COVID-19 vaccine and from Indian manufacturers, Bharat Biotech and Serum Institute of India, and the nature of the vaccine, how it works, the mechanism, that has been asked in the exam there, right? Another question, very different question from biology has been asked from cellulose, right? How and what way cellulose convert into CO2, water and other end products. So that is where it is asked from biological perspective. So what we find last year, Nobel Prize, that blue LED was off the track question. Even this year, biology question is off the track question. So we'll go into the explanation part of each of these three questions. But before I start with that, let me give you a kind of general outline of how many marks and how many questions are being asked over the last few years of the main examination. Let's look into this. So if you see the graph here, and we take the review from 2017 onward till 2022. Now this year 2022, there are three questions are there, which I've just mentioned right now. And the total marks comes 40 marks. If you look at 2021 exam, GS3 paper again 40 marks, three questions are there. If we go back 2020, that was 50 marks question, right? Questions were, four questions were there. But if you just go back here, right, and see 2017, 18, 19, 35, 25, 35, that kind of range of question, two or three questions have been asked. So the point here is that in the UPSC mains exam, continuously three questions are being asked, right, of either 150 words and the 250 words combined, right, mixed type of questions are being asked in the exam. So if you see them according to the syllabus, you all know that the main exam syllabus, if you review, we find that one area is development in science and technology, indigenization, IPR issues, development in space, IT, biotechnology, nanotechnology, robotics, right? So if we look into that, even the contributions of Indians, and we break into the sub components, we find that this year 2022 there are two questions of 25 marks that is based on development of science and technology. That is one is the vaccine development from Indian vaccine manufacturer, and second one that is based on the, if you say that in the biological and part of it, Let's come to that. Now, another one you'll find here in terms of indigenization part, the vaccine part we can take into that, and even the part of the certain thing of energy which have been asked in other papers. That means there's some kind of linkages, right, linked with it, because science and technology is a very dynamic area. Not only GS3 paper, but other papers also questions are being asked. But purely if you look at here, science tech development perspective, only one question is vaccine development program. Biology question and the part of the JWST, they are not from Indian development point of view, right? They are just one global perspective, and the second biology question or cellulose, a general biological question there. So now this is what, but if you go back here, 2021, again, you'll find 3 question development, 2020, 4 questions were there. And we go 2019, 2 questions were there, 20 marks, and the 1 question was 15 marks. If you go back to 2018, 1 question 15 marks, 1 question 10 marks, 2 questions were there. 2017, 1 question 10 marks, and 2 questions 25 marks were there. Overall, it fluctuates around the same 30, 35, 40 marks there. Now, let's look at the question. The first question, that is a question from the biological point of view. It was very unexpected question, right? That normally people prepare biology part in the prelims exam, right? But now UPSC keeps on experimenting. So even the question from prelims concept are now coming into the main examination. So let me read the question first. Each year a large amount of plant material, cellulose, is deposited on the surface of the planet Earth. What are the natural processes of this cellulose undergoes before yielding carbon dioxide, water, and other end products? A direct question here. That means first it gives the idea that large amount of cellulose is made on the earth. Now, what are the natural processes which convert this cellulose into carbon dioxide, water, and other end products? That is a direct question asked in the question paper here. Let's understand here. So we can say that here, so the main aspect of the question is about cellulose and the process of breakdown of cellulose. That is here natural processes. So it is asking not process, but is asking the processes, plural, number of process. So natural processes of conversion of cellulose into the products which are the carbon dioxide gas, then water molecule, and then various other types of end products, different types of end products. Now, this is what the question means. So, a model answer, when a student write answer of this question. So, the very first thing what comes here in the introduction is to write about cellulose. A student has to write about cellulose. That has to be introduced. Then the body of the answer, processes with the end products have to be mentioned here. Now, the body of the answer, then natural processes. Let's say 2 or 3 natural processes which can be outlined here for cellulose conversion. Now, this question is a technical question, and in the answer, biological concept, the process have to be reflected in order to get marks, right? If it is a very generalized answer, marks may be given less, but if it is based on biology and proper explanation based on the concept, then the candidate can get marks. So, if you see here, there is natural processes for cellulose conversion. Now, it is a 150 words question. So, it has to be directly mentioned point wise with examples in the body of the answer. And the conclusion part, which I think is that in this kind of question, conclusion can be a very simple way that same natural processes which break down or convert cellulose into the various products can be artificially or by humans can be converted into lab or industrial processes to yield valuable products, which we are right now doing at a commercial level. That could be a conclusion line to end the answer. Let's look at introduction part first. So, introduction part first here about the cellulose. So, here it can be written here that you all know that you have read in biology that cellulose is a carbohydrate. 4 types of biomolecules: carbohydrate, protein, lipid, and nucleic acid. Cellulose belong to polymer, is a polymer and is a carbohydrate. So, you have to write here the cellulose is a polymer of glucose. The monomer unit is glucose, right? It polymerized, and it is basically a polysaccharide. It belongs to the class of polysaccharide. The monomer unit is that glucose which combines them to form with the branching. It forms the cellulose. So, let's say in the introduction it can be written that there are 2 types of polysaccharides from the plant, that is the cellulose and starch, and both are made from polymerization of glucose molecules which plant prepare by photosynthesis, right? This you are talking from plant material point of view. That's you are introducing in the answer there. Then you are writing here that this is a present in large amount, right? It's a presence is there, right? If you take the presence of such kind of the material, you can give example here like a cell wall. Cell wall of plants is made up of cellulose. Then again, no fibers, right? Fibers, like for example, sometimes the banyan tree you have seen that prop root, those kind of fibers. So plant fibers, right? They are made up of cellulose there. Another aspect in the introduction, what you can write is that cellulose is insoluble, is a insoluble form of a carbohydrate, right? Although it can be hydrolyzed, but normally in water, it does not dissolve, is insoluble and exists as a microfibril, like microfibril and crystalline. It can be microfibril and crystalline. Crystalline form can be existing there. So what has happened here? You have in the answer introduction part, you are writing about cellulose, that you are bringing in a paragraph containing all these points there. At the same time, what you can say that cellulose can be hydrolyzed, right? But it doesn't happen directly. Plants can convert into starch and glucose and use it for respiration to yield the energy. But what is a normal thing if you talk about animals which consume the such kind of materials, plant materials, that's the herbivores, and they cannot directly break down the cellulose. Cellulose can be broken down with the help of enzymes, and this happens by bacteria or microorganisms. So, like here, broken down, like here, it requires enzymatic breakdown into different types of products, right? At the same time, it happens with the help of bacteria. Like animals may have bacteria which can help in the breakdown. So, we are just introducing that microbial breakdown, because such kind of microorganisms contain the enzyme which can help in the breaking down of cellulose. So now what is here? In the first part of the answer, you are writing about cellulose. Now slowly you are moving towards the processes by introducing that the direct conversion which the plant can hydrolyzed into glucose unit, and then it can undergo respiration, where the glucose can be broken down into carbon dioxide, water, and the energy. At the same time, we look from animals' point of view, which consume such plant materials containing such cellulose, has to break down, right? Either using enzymatic process or with the help of microorganism which contains such enzymes can break down. So what is now here, after just mentioning here, let's say 3, 4, 5 sentences, you'll move to the processes now. That's the body of the answer. So the body of the answer, now you have to focus in terms of the processes here. And since question is asking about natural processes to convert cellulose, right? So now here in the answer, you will write that certain type of enzymes, break down cellulose into various end products. And here you can give an example. For example, cellulose breaking down cellulase. One type of enzyme is cellulase enzyme. Is an enzyme which can break down cellulose into its constituent glucose unit. So what cellulose enzyme does? This cellulose can be hydrolyzed, broken down into the glucose, right? Glucose subunit, particularly beta glucose, the monomer unit. And that can be done with the help of the enzyme which can convert into that. And then you know that this can further be broken down into carbon dioxide, water, and plus energy, that is ATP. This happens in respiration. Hydrolysis is part of it. Even normal the plants, right? Even the plant can hydrolyze, hydrolytic enzyme or hydrolysis into their starch. It can convert into starch, and then starch can further be converted into glucose, and then it can undergo respiration process. This I am talking from plant point of view, where plant can utilize cellulose. That could be another way here. Even there are biotransformation here in the nature. There are also biotransformation, not only in terms of hydrolysis of starch, but it can be biotransformation of cellulose into other type of products here, into amylose glucose here, or let's say hexitol, or let's say sorbitol, etc. Even plants, by its cellular mechanism, can convert the same cellulose into other kind of products, which may be very valuable to human beings, like for example sorbitol, hexitol, and amylose glucose. So now these are the naturally done by plants itself. This in the form of plants can be converted, right, into this kind of product. So if a candidate is able to write that in the answer, can bring about this with the example here, what I've written on the board here, right, in the body of the answer, can help in getting marks. Now what is the next level here? Let's say the same cellulose eaten by animals, right? So how the conversion of cellulose can take place there? So another thing which you know that is a very important one, that anaerobic breakdown, right? Anaerobic breakdown of cellulose, and I'm sure you have read this particular thing, that where cellulose can be converted into ethanol, right? Ethanol, that is alcohol, that is using here anaerobic condition, absence of oxygen and anaerobic condition where oxygen is less, and this happens with the microorganisms, microbes such as that there could be bacteria, right? Or I'll give you the example of fungi, the yeast. The yeast is one of that. Then again, it could be done by certain type of other micro like aspergillus. So you can write those example there, which are capable this kind of like your microbial breakdown, and that this is what is called as fermentation, right? The fermentation process which can yield and convert into that. Now this process happens naturally wherever cellulose materials are there, like for the dead plant matter which contain the cellulose and the bacteria in the soil or such microorganism, it is buried into the earth. One process where carbonification happens, where the dead matters get broken down, right, in absence of air and oxygen beneath the soil, right, by such kind of bacteria or fungi into the part of the other material there. So that is also naturally takes place. That you can link with the geological thing where humus and other carbonic matter is formed there. So again, this is again the natural process I am talking about. This is again the one is the fermentation by such microorganisms, and they derive the energy by breaking down the cellulose. That is the part of it. Then that if let's say animal, that animals such as that cattle, right, cows, like for example cows, right. There's even you can also take other example such as not only the cattle like cows here, even koalas. You have heard about koalas, that Australian bushfire. Many of the koalas died. They also, because they feed on this kind of grass or the herbivores like. Then that here another one pandas there. The pandas, China. We have in India red panda. Now those animals which feed on the plant material, they take the plant material, they feed on that. They're herbivorous in nature. Like those kind of organisms. Now what happens is that they have here this kind of bacteria, right? Bacteria present in the rumen, like in cattle, right? Cattle, those kind of rumen and gut of such animals. What happens in there? Where the food, the plant material which is consumed by such kind of animals, that as animals break down, right? It break down to provide the energy there, break down this to yield the energy, and also that it also help in the emission of methane, the methane gas. That the one of the source of methane gas, that is in which is a greenhouse gas, is the animal husbandry. From animals it happens there. Another thing could be here even the plant material which are there into water bodies like wetlands and all, where anaerobic conditions are there in the soil. Again the microbes can break such kind of plant materials of the dead aquatic plants or any such plant which come to such kind of water bodies and wetlands like swamp, marshy land, pitland. Again the anaerobic condition can break down such plant materials and result into emission of methane gas. That is here you are talking about emission of methane gas. Right now about humans, what we consume, we do have bacteria in the body, but those are not capable of breaking down cellulose. The cellulose can be not broken down. So mostly if you take from human consumption, such kind of a plant material, cellulose and fibers, they help in roughage. That helps in digestion part and help in not getting the constipation. That is where the roughage as a part of diet, we take that. So they are also containing cellulose, but human body doesn't have those kind of a system of the enzymes and the bacteria or microorganisms which can break down. So simply get passed out of the body. But if we talk about such animals, they are capable of breaking down using such enzymes or using such microbes which can support in that kind of breakdown there. So what happened here? Now you are writing in the answer. Suppose that you have written around 80-90 words explaining all these processes there. That's what there is a catch in the question. It's not one natural process, but there may be different natural processes where one can write in terms of the breakdown product, right? But what is the main here? If you look at here, the main aspect of here is that where the main aspect which comes here is that if it is hydrolyzed, right, which form the glucose unit, beta glucose unit, 1,4 beta glucose unit, and then it can be converted into the process of carbon dioxide, water, and then the energy, the ATP energy. This is where the plant normally does their natural conversion. This is what happens here. Or let's say this gets converted to starch, and again the starch can be converted into glucose unit. Again, these are natural processes. Or may be converted into ethanol by, let's say, anaerobic process. This happen normal, the anaerobic process ethanol. That can also be part of it. Or the plant can be converting into other kind of end products, other products by using enzymes, different types of enzymes, right? This could other products like I mentioned that hexitol or some kind of other kind of material which can be made there. So this all you have already mentioned in the body of the answer. You have already mentioned in the body of the answer. So that is sufficient. If you are able to mention there, that is sufficient part. Now what is a part of end product? Now when you conclude the answer, right, when you conclude the answer, so here the conclusion part in the answer, what you can do is that simply you can say that these are natural processes which happens, but even we use the similar type of processes in industry. So here, let's say if you look at like that human use industrial processes, right, of biofermentation, where we can convert into many other usable product, valuable products, right, in a bioreactor. In terms of biotechnology and chemical engineering in a bioreactor, right, and one of the aspect of this can be even the bioethanol, which we can use as a biofuel, right. Now today, a lot of research is being carried out to convert such kind of a plant material through the process of fermentation into ethanol, and those bioethanol can be used as a fuel in the vehicles like ethanol blended petrol, or we can use such kind of a material here, like here cellulosic material, that could be converted into the other part of conversion, that is a here conversion into other materials like that today paper, such as paper and pulp, right. Paper and pulp is a very good example where we use such kind of process there. That is again conversion there. Or let's say not only paper pulp, let's say some other kind of a material, let's say gelatin material, right, which can be used there. Or let's say we can convert into the kind of some kind of a value addition medicinal kind of a material there. So same thing what plants can do, we can also try out through research, through some kind of a chemical synthesis or chemical reactions, or using biotechnology method, biotransformation, enzymatic process using enzymes into many other valuable products. Today it is done worldwide. Corn, molasses, bagasse, right? Then again the process of fermentation. There is a process of fermentation where ethanol as a biofuel can be done. So in the concluding part of answer, you cannot write everything. You conclude that the same kind of natural processes today are carried out in industrial processes to convert such plant materials containing cellulose either through fermentation or biotransformation in some other kind of products, and you can give examples. With that, you can end your answers, and you can mention about biofuel, the part of it. So if you look at, if a person, a student, while answering the question, get the framework of the points and proper structure of that and able to write it down, will be able to get marks, right? So this is a very odd one question asked in the exam, but it is not a difficult question. Very basic concept, one can easily write answer. So now what I'll do, I'll go to the next question, that is James Webb Space Telescope. So the question first introduce the background. So let me read the question here. Launched on 25th December 2021, James Webb Space Telescope JWST has been much in the news since then. Current affairs, right? What are its unique? Now, what is very important? What are its unique features which make it superior? Now, it is a comparative features which make it superior to its predecessor space telescope. If we take the other space telescope, right, before JWST, James Webb Space Telescope, how this JWST is superior than the other beforehand? So that is what is asking here. Now the second part of the question, and what are the key goals of this mission? Main goals of this mission. Only the important goals have to be written. And the last part of the question, what potential benefits does it hold for the human race? So what potential benefits does it hold for human race? And one has to answer this question in 250 words. Right? Let us go to the model answer here. So the model answer is like this. Introduction. In this question, when you start writing answer, already the question has introduced about the date when it was launched. So you can say that about, you can talk about this telescope, that is a James Webb Space Telescope, right, named after. If you know this information, you can write the named after NASA writer, former director, that is a James E. Webb, right, who served from 1961 to 68, right? In a one sentence, you can write that is an advanced astronomical telescope. Is an advanced astronomical telescope, right? Then it's a predecessor. Now, then it's a predecessor. When you're writing here, space telescope, you can give the examples of the predecessor space telescopes, Hubble and Spitzer. So one is Hubble Space Telescope. 1990 NASA American Space Agency which used there, but now Hubble has been retired. There's another one called Spitzer Space Telescope. Then you know that other space telescope which has been there, Kepler, to find out exoplanet, Kepler Space Telescope, right? Then wide area telescopes, you can give that part. Now in the answer, you will also write about where it has been placed there. So you can also write that JWST has been placed in a halo orbit. Now it's a very important point here. Halo orbit. In a halo orbit, that is about a point orbit around Lagrangian point L2, right? Lagrange point. So Sun Earth Lagrangian point L2 at 15 lakh kilometers or 1.5 million kilometers away from the Earth. 15 lakh kilometers, right, away from the Earth. So again, you can write the position of that. So you can write the orbit around Lagrangian point L2 of Sun-Earth system, right? Sun and the Earth. If we take that 5 Lagrangian points, the L2, the backside of the Earth, right? L2 at the 15 lakh kilometers away from the Earth, using France Ariane V rocket, Ariane 5 rocket. It was launched using Ariane 5 rocket from where? Kourou, French Guiana, South America, which is France rocket. So using that, it was launched to the L2 point. That is if we look into this part, L2 point, that is a part of here, Sun and the Earth. And those any candidate want to draw with image can also show that. So here L2 point is here, the back side of it. And there in the halo orbit, JWST has been placed. This has been launched, right? And that distance here is around 1.5 million kilometer or 15 lakh kilometers. Right? So Sun Earth system is done there. And for launching, NASA used the Ariane 5 rocket of France from Kourou, French Guiana. Now what comes here? This is a collaborative project here, collaborative astronomical project. So you are writing in the introduction about the 3 main points which I am writing here: astronomical project of American Space Agency, NASA. Then comes ESA, European Space Agency, and Canadian Space Agency, CSA. So European Space Agency and Canadian Space Agencies both helped in the design of the instrument, cameras, the mirrors, and the development of the entire telescope. So these are the joint mission here. Since question about the mission you are writing about in the introduction, it is a 250 words. So you have a liberty to write more points there, right? So this you can mention to that. Now coming to the body of the answer. Now what comes here? Body of the answer. Like when you come here, body of the answer. Now body of the answer, first part is that features. Is unique features which distinguish James Webb Space Telescope from its predecessors. And one of the very important predecessor is Hubble Space Telescope, right? So here what you can say that the first thing what comes here, unique feature, either you can write in a paragraph or you can write in point wise. So I'm giving here the explanation point wise here. So let's say unique feature. The first thing is that it has 18 hexagonal mirrors, right? 18 hexagonal mirrors, right? Coated with gold coated, is a gold coated with beryllium. Now why it has been used? Because the main purpose of this telescope is to look into the universe, even going back in time, what happened after Big Bang, the origin of the universe part of it. So in order to get protected from the sun and the glare and the heat, that's where these mirrors are there, beryllium being used there. Another very important thing is that it is a basically, if you look at size, right, it is a 26 meter, 26.8 meter, that's big here. So here around 26.8 meter, that is around 26 meter, right, size. If you look at Hubble was having only around 2 meters. So it is almost like 10 times bigger So you can say here the 10 times bigger. The mirror is 10 times bigger than Hubble. Then Hubble's mirror here. Right? Now I'll give you another thing here. Hubble was placed in low Earth orbit, whereas JWST is in the L2 point, Lagrangian point 2, where hello orbit. Another major, but feature wise, if you look at the mirrors, is all the mirrors combined that bigger kind. This is a much bigger than Hubble's mirror. Now what is here? Another very unique feature is that it is for infrared imaging. So here near infrared, right? Then mid infrared, the observation which is going to make, and then far infrared. And along with this, not only infrared here, but also it can have observation in red and orange. That part of the visible light, part of a visible light spectrum. Whereas if you compare with Hubble, right, spectrum here, so Hubble, if you look at, Hubble was also infrared, but it was not that much, more optical, visible, and but here near one. That means even a grain of sand, very small, JWST can get a very detailed the information about anything in the universe. So it has got a high resolution, right? It is high resolution imaging there. And not only this one, it can also, its instrument can be inclined, like here 85-degree angle inclined, and it can be observe even solar system and the Kuiper belt objects. So these are the features part of it. It has got the cameras like near infrared camera, mid infrared cameras, then near infrared spectrograph. These are instruments. If you see the instrument here, that is a payload, but is the part of this. So here near infrared, this the imager or camera. Then it has here near infrared spectrograph, which it carries as a for making such kind of observation. Then it has a mid infrared camera. So the advanced instrument, at the same time the mirror. What is very important is the larger size mirror, right? Which can capture all the infrared radiations and all, and is able to. It also minimize the reflection. That's why the coating is there, gold coating there, reflective part of the mirror, and the infrared and getting the information about the universe, deep universe. That makes its a unique feature. And compared to Hubble, it has a larger mirror, then better in terms of the instrument and the cameras and the infrared part of the spectrum and red and orange part of spectrum of visible light. It can capture all the data in the image. So this part of the answer, one has to write about unique feature and compare with the previous one, the Hubble. What I put across the point here. This has to be explained in the answer. Now what comes here, the next part of the answer here can be here, that is a goal of the mission and key goals, the important goals of JWST mission here. So it can go back in the time. So first here, that is this observation of first galaxies. Right, first galaxies that formed after Big Bang. It can collect all this the data there. Then again here, observation of formation of the galaxies, the normal galaxies where the observation can be there. Then observation, right? Observation of formation of stars, you know that nebula, gas and dust of stars and planets. And in this context, what was the headline? What made the headline? What was in the current news? That was Carina Nebula. JWST has looked into this nebula, Carina Nebula. The first image which was brought, which has so many galaxies and everything there, that was brought by the camera of the JWST. That is again the nebula, star formation, planet formation. Then again here, it also can have observation of solar system, right? Or you can say here exoplanets. That is the same what Kepler has found, Goldilocks. Those planets which is similar to the Earth, Earth-like planet, exoplanet which may have atmosphere, water, right? That can be found out. Then again here, it can also observe observation of a solar system. Now, if it is a tilted, right, our own solar system, if that mirror and the telescope is tilted by 85 degree, where it is the back side of the Earth, tilted by 85 degree, it can have close observation of Mars, Jupiter, Saturn, Uranus, and also Kuiper belt, and you can get more detailed information. So here, observation solar system, detailed study of Mars, Jupiter, right? Those kind of planet, and also Kepler Kuiper belt object. That's what this telescope has a lot of significance, and also Kuiper belt. That means if the NASA control and then tilt the telescope in such a way 85 degree with the angle, then it will be able to even see through the solar system and get all the information there. That's why this is a, can say, very advanced kind of telescope. See, if any candidate write point wise all the goals, will definitely get marks, right? Then what comes here, not only this one, what I'm now bringing here, the how it will help the mankind here. Like the last part of the answer is that what benefit, like here, what benefits to human race because of this particular James Webb telescope and the study of this, how the human race will get benefited. So you can write there that all the information, data, and about this. The first is this one, that it will understand here. Then in terms of astronomy, understanding of the origin of the universe. That is what you know, that in terms of the mystery which is there, let's say here, in terms of the Big Bang, it will give all this information. Early part of the universe can be studied there. But you still wonder how this human race understanding that, because we should know how things have started. That's a astronomical knowledge. The knowledge will be there. Then most important from human race point of view, about the universe, that is a astronomical research about galaxy, or let's say about a nebula, about star formation, about this kind of planetary formation. But how the humans will get benefited in that context, I can tell you here, that is one of the most important thing here, this one, finding this exoplanets, right, or Earth-like planets. What Kepler has been doing that, right? But what Kepler has done there, or you can say search for life, search for life, search for life, finding out ki are we the only one or are there? And another very important here, this one, study of solar system, study of even the solar system, right? We can study here the evolution, how the solar system is evolved, evolution of the planets, right? Are there any other body, let's say here, finding new celestial bodies? Even in our own Kuiper belt, there are certain type of bodies, the celestial bodies. Outer edge of the solar system, finding out, studying about outer edge of the solar system. So again, these are goals of this mission, but again, what it can help? Maybe we can find out minerals, mineral composition of the, let's say, the planets there, and other things here. That again, looking for life, hunt for life, search for life, looking for water, air, atmosphere, other planets, other our nearby solar system, or the galaxies. All those understanding will be there. So when you say human race, we're talking about human race. The one is that in terms of knowledge, where we stand in the universe, understanding mysteries of universe, all the theories of astronomy. But again, in our solar system, understanding how Earth has been formed. Our Earth, which has evolved, that similar thing, formation, other kind of such exoplanets, that will enhance our understanding of how the life, and maybe the other life may be searched, that could be there. Right. But at the same time, whatever the finding, like minerals or whatever maybe in our own solar system there, that can help for the future, not today, maybe 100 years. Science is always for which looks into not only development and benefit of present, but also for the future of the mankind. So from astronomical point of view, it is going to enhance that understanding, and with that you can conclude the answer, right? That with that answer can be concluded. It can enhance, help further more research to develop some new technology, and those technology can also be there in the industrial, like for example, all the instrument or whatever has been developed, it has led to research, and let's say similar type of instrument or such kind of thing, component, cameras and all, or mirrors, could find some other application on the Earth. So that is the economic point of view things linked with that. That also you can highlight there, right? So 250 words, you can write a lot of things. That's why UPSC gave 2-3 parts of the question to be written. Right? That's what is given there. So if a candidate has written all these points one by one in a proper manner, would get marks. Right? Now let me go to the last question here. So I am going to the last question here. This one, what is the basic principle? See here, what is basic principle behind vaccine development? Now this question is from biotechnology. Now how do vaccines work? How do vaccines work? Second part of the question. Third part of the question, what approaches were adopted by the Indian vaccine manufacturers to produce COVID-19 vaccines? So the question is divided into 3 parts, and one has to answer write the answer in 250 words. So again, you have certain words to write it down. First is that basic principles. By basic principles, right, of vaccine development. So this is the biotechnology, how the vaccines are made. Second part of the question is asking how the vaccine work, right? How vaccine work, the mechanism once we take vaccine, how it work inside the human body. Inside the human body, how the vaccine work. And the third part of the question is that about the Indian manufacturing companies. You can take 2 examples here. One is a Bharat Biotech with support from ICMR, Indian Council of Medical Research, Bharat Biotech India Limited, that is in Hyderabad based company with ICMR. This was, you know, that Covaxin. And there is another company which you know, that is a Serum Institute of India. That is a Pune Serum Institute of India, right? But it is a collaboration with Oxford AstraZeneca, SIA, which has developed a vaccine, Covishield. Although there has been also development of vaccine manufacturing by licensing, like 𝐾𝐫𝐢𝐬𝐡𝐭𝐢, Russian Sputnik V vaccine, Dr. Reddy's lab, right? Cipla, again in terms of this, Corbevax, now as a booster dose. So now Corbevax as a booster dose, that is a part of it. So there you can write in that. And if you see, not this year prelims exam 2022, but if you go back 2021 prelims exam, already a question in objective type has been asked in the prelims exam. So now you see the question in the prelims exam about the same vaccine and all this one, which was asked in the exam, and then now in the main exam in a different but touching upon the same idea, mains question has been asked. So now if you look at here, I'll go to the answer here. Let's look at here the first introduction. So introduction of the answer. What do you have to write here? That this one, you can write about biotechnology research. Like what you can start with that? Biotechnology is an integral part of human development, and the development of vaccines, drugs, different type of therapies help a human being fight diseases. Right? You have written as a sentence. Then you write about vaccines help in providing. It helps in providing the preventive healthcare. Preventive healthcare. Right? Preventive healthcare there. One is that a person get disease, take medicine, drugs, or any kind of treatment, therapy to recover from the disease or illness. Second is a healthy person, in order to get that kind of infection or disease, to protect the body from there. That is a enhancing immune system. So what here comes here? So what vaccine does? The vaccines help in enhancing body's immune system. Right? Body's immunity to fight diseases. One comes a natural immunity, which we acquire from the birth or after infection. But another is that which we acquire or get through the vaccines. So in the introduction, we are writing that biotechnology is an important area of technology where the therapy, medicine, drugs, vaccines are all developed. There then you can write that India is also one of the leading countries in the world in terms of biotechnology research and development. Then what you can bring here? Then you can bring in the answer that vaccines are part of preventive healthcare. It helps in boosting the immunity to protect from getting a disease. Right? That part you are writing there. Then after that you are writing that there are different types of vaccines. Right? You are writing here that you introduce that different types of vaccines are there. Different types of vaccines used in modern times. And here you can give examples here. Like here, inactivated, or what is called here killed pathogen, where the virus is killed. Those kind of vaccines are there. Or one also use a recombinant vaccine here, where the genetic engineering, recombinant DNA technology is used to develop the vaccine there. Or now messenger RNA, mRNA kind of vaccines have been built in, which is also used there. And you can also write in the part of the initial part that vaccines are administered, right? Vaccines are administered. One is that intravenous, administered intravenous. Now when you say intravenous, that is a blood injection. Then oral route. Now you know that oral route, that is a pulse polio. Polio is given as a drop also. Polio like now IPV given through injection also there as a jab. Same here, other vaccines are given. Hepatitis or any kind of COVID-19 also comes there. Another now comes a nasal also there. Nasal route. Now you know the Bharat Biotech. It was in current news. Even in the answer you can write about Bharat Biotechnology development of this vaccine which is nasal route. It can go into the blood and the stomach and then and produce antibodies in the human body there. So there can be different type of mode of getting the vaccine into the human body. That also you can write in the answer there, introduce in that part. Then what comes here? Then in this context, when it comes to body of the answer, the body of the answer, now in the body of the answer, what you can do is that the answer is asking, the question is asking there about the how do vaccine development here, right? How now the question is about vaccine development. First part comes to vaccine development. So what you can do in this vaccine development, you can write in a paragraph. Although I am giving you the points to understand and write an answer, but you can write in a paragraph, or even if you want to write in points, you can do that. So here what is done here? So here either like here first is that type of vaccine. Okay, what type of vaccine we want to develop, right? Whether we are using a killed virus, killed pathogen, right? We are killing the pathogen completely. That is what is vaccine. What is the vaccine here? Is a sera. Is a sera. Is a serum which contains there killed pathogen. Or we are making which type of vaccine? Are we not only if we want to make a vaccine, now vaccine could be also here anti-sera. What is anti-sera? That is a blood serum, that is after the blood serum containing antibodies, which the person get this one, so directly they can get antibodies inside the bodies through the vaccine, and those antibodies can fight anti-sera. Or maybe here the vaccine may contain here protein subunits of the pathogen. Let's say here the COVID-19 virus here, COVID-19 virus or the pathogen, like in COVID-19 spike proteins, which are been, now this is developed, any sub kind of component which are developed. So what is done here? One is that we kill the pathogen, that is called live attenuation, live inactivation. In the lab, what we do, we attenuate it, we inactivate the whole virus, and that is the solution, serum, becomes the vaccine which one can take. So here to talk about methodology. Here the question asking the method. What are the method of development of vaccine? Or what we can do, we can anti-sera. What is here? In live of the virus here, let's say pathogen, and then that is prepared. Here what we do, we immunize animal, and animals with that virus there or the pathogen. Now animal will produce antibodies, and here collection of antibodies. There we take the antibodies there, collection of antibodies. Or we can take the protein subunit. Now from where the protein subunits are made? This is made by genetic engineering. We do the genetic engineering or recombinant DNA technology. That is or what you call recombinant DNA technology. Recombinant vector. We use the recombinant DNA technology method to develop that. Recombinant DNA technology. What we do, we genetically engineer a bacteria with a gene which make that proteins of the virus. Let's say the surface protein of the virus, original virus. Suppose that's a COVID-19. Those protein, that gene which make that virus, we are using recombinant DNA technology or genetic engineering. By that we get into the bacterial plasmid, and the bacteria make those kind of protein, we take it out, and that becomes a vaccine which we take into human body. So you are talking methods, how we can develop the vaccine. So here method made by genetic engineering, right? Genetic engineering or recombinant DNA technology to develop, right, proteins, surface proteins that act as an antigen that elicit immune reaction in the human body, and we produce antibodies. So those proteins of the virus or the pathogen that genetically engineering and the recombinant bacteria, just like normal we make insulin, the recombinant insulin, same method done in the lab to develop those protein, and this protein has become part of the vaccine. So again, that is the method, or the method could be here messenger RNA. So messenger RNA of human being, mRNA, right, or which contain here with RNA of the virus. Let's say RNA of the COVID-19 virus. And what mRNA does, it provides template. So imagine that the COVID-19 virus, the proteins on the virus, and those protein which is made through the gene or the DNA, and the information coded in mRNA, messenger RNA, which normally inside human cell, where transcription happens, mRNA reads the gene or DNA in the human body, and then translate and make protein. Same mRNA of the virus which make the virus protein, that mRNA with the human mRNA taken as a vaccine, this mRNA help in building the protein of the virus by the cell of the human body itself, and those protein become antigen, which really produce antibodies by our WBC, our immune system. So if original virus comes, the antibodies. So here is Moderna. Moderna has this kind of a method here, where protein subunit and the part of it is part of Serum Institute of India, Covishield, that is got. Covaxin is this one, killed virus, right? So you are explaining the method here with RNA of the virus, messenger RNA of the virus, to synthesize the protein subunit of the virus. So what you have done, you have in a proper way, paragraph wise, you have explained in your answer how in the lab which methods are used for making vaccines, right? That's a part of it. Then what comes here, the next part of the answer, that is how the vaccine work. Like here, so when we take vaccine, right, taken inside the body. Now you are explaining in this area inside the human body, either the vaccine contains the dead virus, right, or let's say it contains the protein subunit of the COVID-19 or messenger RNA, so that elicit immune response to produce antibodies. This is again biology part, what you are aware of, antibodies specific to, right, antibodies produced specific to antigens. Let's say here the antigens are the surface proteins of the virus. Antigens, surface proteins of the virus, let's say COVID-19 virus, right, or any other virus which we make vaccine there. So immune response to produce antibodies. You can even underline those kind of sentences that antibody specific to the antigens, surface proteins of this, by the WBC, by the white blood cells of our immune system, of the immune system. So once we take the vaccine inside the body, so our WBC lymphocyte, right, produce a immunoglobulin, that is antibodies. That antibodies are specific to the surface protein of the COVID-19 virus, and that is there in the body. So now what happens here, when actual infection, right, when actual infection takes place, let's say when the virus enters the body, so these antibodies, right, which have been produced through the vaccine, so these antibodies fight and kill the virus, the virus which enter the human body. So this is you have written in a general term, right? Now you can ask the question, you have to write biologically and explain some specific term? Yes, if you can do, well and good. But if you are paucity of time and you are not able to, at least the basic inherent mechanism, you just write it down so that you can get marks. So here, So let's say this antigen protein, either if you take the 𝐃𝐫𝐢𝐬𝐡, the subunit of the virus, or the dead virus, it also has a sub protein there, or mRNA that makes the protein. Ultimately those protein become antigen, and our WBC recognize them, make antibodies. This is how the immune process get working in the human body. So you have explained that. Which again the real virus comes, it fight and destroy the virus. So here the next part of the question. Now comes the final part of the question, that is the Indian manufacturer, like Atmanirbhar Bharat, Make in India program, and India's fight against COVID pandemic, COVID-19 pandemic, Indian vaccine manufacturer. Now here you have to write about the two types of vaccine and how they differ, and their mode of mechanism of infection that you have to bring. So what you can do here, in paragraph you can write, or in a tabular form you can explain that. Either you can take in paras you can write there, or you can take into the tabular form, you can distinguish the type of vaccine, or you can write point wise. Any of the method you can follow. So what I'll do here, I'll just make a tabular form. In the answer, you can use any of this kind of method, either write point-wise differences, their mechanism, or write in paragraphs, or you can use table. Let's say here, one is this 𝐃𝐫𝐢𝐬𝐡, and the another is COVAX. So you can write there that this is by Serum Institute of India, right, with AstraZeneca, UK-based AstraZeneca. That is where the collaboration is there. This is, you can say about Bharat Biotech, Hyderabad-based company, Bharat Biotech India Limited, with ICMR. Now, what is the important part of the question? The differences. So, what is the difference here? It used the chimpanzee. Right, adenovirus. This virus is not the real COVID-19 virus, adenovirus, which infect chimpanzee, not humans. So will not cause any problem with the human, but chimpanzee. Adenovirus as vector, it becomes a vector, right, carrying protein, the protein subunit of COVID-19, protein subunits of COVID-19, that is a spike protein. That has a spike protein, right? The spike protein, that is a spike protein of COVID-19, that is spike proteins. This is where it is coated on the adenovirus, right, which infect chimpanzee. That is a carrier vector. Whereas here, what is there? It is inactivated or killed whole this COVID-19 virus. The real COVID-19 virus has been killed, and that is taken as a vaccine. Whereas this one, what we do, we have taken the adenovirus, that is the adenovirus, it has an RNA here, and this adenovirus with DNA and the protein which it carries. So this protein has been added through genetic engineering or recombinant proteins here. So these are the spike protein that depends on the variant of the COVID-19, different variants, you know that. And then this carrier is adenovirus. Which adenovirus? Not human adenovirus, chimpanzee adenovirus. So that becomes a vector, right? When this virus will taken as a there's a 𝐃𝐫𝐢𝐬𝐡, this adenovirus as a carrier will carry this spike protein of the COVID-19, and that our WBC will recognize and make antibodies. So when the real virus COVID-19 comes, antibodies will fight. But here, what we do here we are taking this, the exact, here we are taking the killed, this here is a killed COVID-19. We are taking the real COVID-19 virus here. That means it's not active, dead. So our immune system recognize, make antibodies. So when the live virus comes, those antibodies will fight. So here it is you can say it's a recombinant. It uses recombinant technology. Is a recombinant vaccine. And this is the like a normal kind of non-recombinant normal virus, non-recombinant, right, vaccine is there. Simple dead pathogen is used, just similar to polio. We use this kind of method, what Bharat Biotech has done. If you compare this Covishield with Sputnik V of Russia, right, Sputnik V Russia, which is made by Indian manufacturer. So in that case, instead of the vector being chimpanzee adenovirus, in Sputnik V vaccine, it is a human adenovirus carrying the spike protein of the COVID-19. So it is also a recombinant vaccine, the Russian vaccine there. The difference is that the adenovirus there is human. Here in the Covishield, it is a chimpanzee. Right, so that you have to write. Now you can also write that now the booster dose Corbevax, which has been brought to the India after 2 doses and 3 1. So now what you can do, you can, after explaining all this in the form of this, now the next level what comes here, that is your conclusion part. I hope this is clear to everyone, the way you will approach answer writing there. So let's now here, so one can write here conclusion. Now what could be the conclusion here? That you can write in that here that India has been successful not only in developing indigenous vaccine, successful in developing, right, intravenous, like by using into blood, right, indigenous vaccine, right, intravenous, you can also write, and now or nasal route, right, route indigenous vaccines, right, but also like doing research, doing R&D to develop the new vaccines for not only COVID-19 virus, not only the emerging, let's say emerging or any new variant, emerging variants of, you know that variants, new type of mutants on the spike protein, variants of COVID-19 virus, but also for other diseases. Let's say, you know that monkeypox, that was in the news, but also other like diseases. And one example you can say that, for example, monkeypox, which is right now in the news also there. So our biotech technology and in make of vaccine, India is one of the leading producers of vaccines in the world. Even you can mention that during the COVID, not only our citizens got immunized, but also we exported vaccine to neighboring countries, other countries. So we have gained an edge in the vaccine manufacturing. So India, you can end your answer that India is today a leading producer, not only research but leading producer of vaccines in the world. Leading producers of vaccines in the world. So that could be your ending statement, which can bring the positive aspect of our development of biotechnology, particularly medical biotechnology part of it, right? So since questions are asking about Indian manufacturer, right? One can write about mRNA vaccine also as a Moderna has got, and those, and this you can write in the introduction part. But when you are doing comparison, you are talking about the Indian companies here, right? And then you are concluding with the ending part that we are not only this but new vaccine also there. We are have that kind of advantage. Then also in the case of we are making manufacturing and also exporting there. With that, the answer can be ended. 250 words, you can answer. You can end the answer, right? I know that I have gone one by one, provided you number of information. So all of you who are watching right now, can do one thing is that wherever you missed out, this video is with you. You can go to that part of the video, can pause, and if you are looking to the points which I have written on the whiteboard, you can write it down, right? Like particularly for those who are going to appear exam in future. But those who have already done the exam, written the exam, so they can at least get an idea what they have already done in the mains exam and where their answer stands there in terms of self-evaluation, right? So we at Drishti IAS and the team of Drishti IAS, right, come out with this kind of explanation for each of the subject to help those who have written already the mains answer to get a good idea about their answer written and how much they can expect in the exam to get marks. At the same time, those candidates, right, future candidates who are still trying, who have not qualified the last year or this year and even trying for the next years, or the new people who are preparing for UPSC exam, they get to know the model answers and the approach to write answers in science and technology, right? So I am really very glad that you all are present here and watching this video. So I thank you on the behalf of Drishti IAS team, and I wish all those candidates who have appeared this year mains exam, written the answers, a very good luck. At the same time, I wish all the best to all of you who are going to appear in the coming year exams. Anything, query or doubt or anything you read, you can always feel free to approach Drishti IAS. We are there to help you out, guide you there. So I wish all the best. Thank you. Take care. Bye-bye.